So we're doing the Krebs cycle. This is also your TCA cycle. So we said after the glycolysis, we had the fate of pyruvate, where we were deciding if there was enough oxygen for the pyruvate to enter the mitochondrial matrix and get participated in the Krebs cycle, basically participate in the Krebs cycle. In order for the pyruvate to participate in the Krebs cycle, we were converting it into acetyl-CoA. One glucose molecule going into the glycolysis was giving us two pyruvates, and then we were using that big pyruvate dehydrogenase complex to convert the pyruvate into the acetyl-CoA. So two pyruvate go in and two acetyl-CoA come out, and then these acetyl-CoA go into the Krebs cycle. The eventual fate of the Krebs cycle is to generate energy, and energy molecules are basically your NADH, FADH2, and GTP. So all these three this NADH, this FADH2, and this GTP are all energy efficient molecules. They are a lot of they're like these energy little pumps that help you stay alive. And then these energy molecules go into the electron transport chain, which we will cover after the Krebs cycle, and generate ATP. So the cycle itself produces a very little ATP, but it produces other molecules like NADH, FADH, and FADH2 and GTP that eventually go into the electron transport and make ATP for you, right? Its real purpose, the Krebs cycle actual purpose is to create NADH and FADH2. And those NADH and FADH2 get feeded in the electron transport chain later on, right? So where does it happen, this Krebs cycle? Can you see this beautiful mitochondrial matrix that I've tried to create using AI? So this beautiful mitochondrial matrix, this is where the Krebs cycle is happening. And I've put these little bows on every step of the Krebs cycle. So we're going to call the Krebs cycle cute Krebs, right? So the cute Krebs happens inside the mitochondrial matrix. Exception is the succinate dehydrogenase. The succinate dehydrogenase step is special because it is embedded in the inner mitochondrial membrane because it is also part of the complex 2 of the electron transport chain. This is something we have to touch upon because the examiners love asking this question. There is a inner mitochondrial matrix not shown here. Here you can only see the mitochondrial matrix, and that is where the succinate dehydrogenase steps happen. And that succinate dehydrogenase step is common. That the succinate dehydrogenase is basically common in the Krebs cycle and also in complex 2 of the electron transport chain. And the examiners love asking this. Okay, so what enters? Only let's enter the Krebs cycle. Let's deepen. Let's jump in. Okay, so you can see this cycle here, right? So this acetyl CoA, this is basically a two carbon compound, right? And it's coming from your pyruvate dehydrogenase complex, taking the pyruvate and converting into acetyl-CoA. It combines this acetyl-CoA basically combines with the oxaloacetate, which is already being regenerated using the Krebs cycle. It combines with this using the citrate synthase enzyme. It throws the CoA enzyme, which is attached to the SH group, the sulfur group, right? And it becomes the citrate. So now you have a six carbon compound. So this first step is just the acetyl-CoA being attached to the oxaloacetate, throwing the CoA SH out and becoming citrate synthase. Now, what does one turn of the cycle produce? One turn of this entire Krebs cycle we've done, it produces three NADH. Can you see it here? Let me just zoom out a bit. Can you see that it produces three NADH, one FADH2, and one GTP, which is equivalent to let me pick a pointer here. Okay. So we're creating three NADH molecules, one FADH2, one GTP, which is equivalent to one ATP, and two carbon dioxide molecules. This is every cycle of Krebs cycle produces this, okay? So this is something I want you guys to remember. Now, this oxaloacetate is regenerated with every cycle of the Krebs. So every cycle reproduces this oxaloacetate which then combines with the acetyl-CoA coming in, which is basically two compound. Oxaloacetate is four compound. Four compound, I mean two carbon, four carbon, right? So two carbon, four carbon, they will both join and create this six carbon compound, which we call the citrate, right? Okay, so that's your, we talk about the energy yield. Just a little bit heads up, one NADH. Every NADH produces 2.5 ATP. Okay, maybe they'll ask you this on the exam. And every FADH2, each FADH2 produces 1.5 ATP. So every NADH is 2.5. per molecule, right? If you multiply it by the three, I am so bad at using this pen, I'm not writing it down. So every NADH is basically, let me just write the text here, okay? So every NADH is basically 2.5 ATP per molecule, right? And every FADH basically is what I just told you, it's 1.5 ATP per molecule. So how much, and one GTP is basically one ATP. So how much is 2.5 multiplied by 3 is your 7.5, and then you have this 1.5, then you have this 1 ATP. You add them all together and you get a total of 10 ATP. And they love asking this. They love asking that you basically create 10 ATP in every Krebs cycle. This is like their favorite question, I think. I've seen it happen like asked so many times. Okay, moving on. So per glucose, we said 10 ATP. Sorry, per cycle it's 10 ATP. But since we're using one glucose in the glycolysis, and that is creating two pyruvates, and two times the Krebs cycle is happening for every one glucose molecule. So that gives you a total of 20 ATP. Right? I hope that's easy for you to follow. Okay, so think of the oxaloacetate. I've explained it to you as a reusable docking station. It comes off as regenerated in the end of the Krebs cycle itself, right? And then it was giving us the citrate synthase, right? The citrate synthase was giving us citrate. Now, this first step basically produces no ATP. No ATP, no NADH, no carbon dioxide. The only thing it gives off is the coenzyme A and the SH group attached to it, right? Because the enzyme now, how is this regulated, this step? This enzyme, this citrate synthase, this enzyme is inhibited by high ATP. So whenever there's high ATP, you don't need more Krebs cycle to go on, right? So it will automatically stop it from happening. Also, high NADH, high citrate, these high energy level molecules all automatically inhibit the citrate synthase. So the first step doesn't happen. Why? Because the cell already has enough energy, right? So when citrate accumulates, it leaves the mitochondria and goes into the fatty acid synthesis, right? This is something you should remember. So when there's a lot of citrate, what happens is it stops this first step from happening, right? And where does this extra citrate go? This extra citrate goes into the fatty acid synthesis and it makes fat, right? So starvation is actually sometimes good for you. You should definitely practice being a little hungry, guys. So this examiners love asking this. Examiners love asking that in a fed state, in a well-fed state, you produce insulin, right? And the citrate is exported into the fatty acid synthesis and it's going to create fatty acids in the lipogenesis pathway, right? So citrate, basically a lot of citrate inhibits glycolysis. Citrate inhibits the phosphofructokinase 1 of the glycolysis pathway, meaning the cell says, I already have enough energy, and glycolysis also slows down. Also the citrate Okay, that's enough about the citrate. I have to move to the step two, right? We've done with step one. Okay, so step two is done by the aconitase enzyme. What is the step two done by? It is done by the aconitase enzyme. Do you guys want to revise a little bit of the step one? The step one that we just did. I made a note about it and I forgot that I didn't practice it with you guys. So this is step one, right? We said the oxaloacetate and the acetyl CoA join to make your citrate. The enzyme is citrate synthase. It is activated by low energy levels like ADP. Whenever you have ADP, it means the body has low energy. It's activated by it, and it's inhibited by all these enzymes that have high energies like ATP, NADH, citrate, succinyl CoA. Right? The USMLE loves asking you that the citrate activates acetyl CoA carboxylase, which leads to fatty acid synthesis, right? The examiners love asking you this question. And high energy means there's no need for the Krebs to continue. Acetyl-CoA has two carbons. Oxaloacetate has four carbons. Together they create the six carbon citrate. You can pause the video here and go over this if you want. Clinical correlates is that the citrate accumulates when ATP is high. Citrate leaves the mitochondria and it's used for fatty acid synthesis. This is how high carbohydrate meals lead to fatty acid synthesis. So high energy, stop the TCA, make fat. Low energy, run the TCA, make ATP. which is why sometimes it's very good for us to go on low energy. So back to our main Krebs cycle, right? This is our main Krebs cycle. We've taken the acetyl CoA and we converted it into a citrate, which was a six carbon compound. We now need to make an isocitrate, which is also a six carbon compound. And in order to do that, we're going to use the aconitase enzyme. So step two, here is our step two. Can you see this? Okay, so step two is basically the citrate. We convert it into isocitrate by the enzyme aconitase. This is a dehydration reaction, and because it's a dehydration reaction, we're simply removing the water from the reaction, right? So the reaction goes from dehydration to rehydration. First, we remove the water and then we add the water, right? First, there's a dehydration happening and then there's a rehydration happening. This simply rearranges the OH group, right? It basically simply rearranges the OH group. There's no ATP produced, there's no NADH produced, there's no carbon dioxide produced, right? One thing I would like to add in here, whenever we have fluoroacetate poisoning, whenever we have fluoroacetate poisoning, it inhibits aconitase enzyme, right? The TCA cycle drops and ATP production falls. It's seen in rodenticides, right? So the fluoroacetyl CoA, the fluoroacetate is converted into fluoroacetyl CoA, which can convert into fluorocitrate. You don't need to know this, but you need to know that the fluoroacetate basically inhibits aconitase enzyme. The result is that the TCA stops, ATP falls dramatically, right? Also, aconitase contains iron sulfur center, therefore oxidative stress can damage it. Remember this. Maybe they'll ask you on an exam or not. I'm not sure, but this is something I want you guys to know. All right, so let's move on to step three, guys. So now we've converted the acetyl CoA into citrate, and then we convert it into the isocitrate. Now we need to take the isocitrate and convert it into alpha ketoglutarate. This is our step three. This is done by isocitrate dehydrogenase, right? So, and during the isocitrate dehydrogenase working on the isocitrate to be converted into the alpha ketoglutarate, we are using the NADH and we're adding a hydrogen ion and converting it into carbon dioxide. This is the first oxidative decarboxylation, meaning oxidation is happening and carbon dioxide is removed. Whenever we remove carbon dioxide, we call it decarboxylation. And whenever we're saying it's something is being oxidized, we're basically taking an energy molecule, adding a hydrogen ion to it, right? And product is basically your NADH molecule. Okay, so the products are you're getting an NADH out of it, right? And you're getting a carbon dioxide removed from it. This is one of the most important regulatory enzymes. Your isocitrate dehydrogenase is one of the most important regulatory enzymes of the Krebs cycle. So let's go ahead and understand the alpha ketoglutarate enzyme. Okay, so this is uh, isocitrate dehydrogenase. Step three, the isocitrate is being converted into alpha ketoglutarate by the enzyme isocitrate dehydrogenase. Right? Now, just remember that this enzyme is activated by high levels of ADP and calcium, which means ADP, I always told you, whenever you have ADP in a cell, it means that the cell is low on energy. So whenever there's ADP or calcium, basically you need to activate the alpha keto, the isocitrate dehydrogenase to convert the isocitrate into alpha ketoglutarate so the Krebs cycle can keep going, right? Why calcium? Now you understand that the ADP means you have less energy, but why calcium? Because when muscles contract, calcium rises. When muscles contract, calcium rises, and whenever there's so much muscle being contracted, you need ATP, right? So the TCA cycle speeds up. So that's something I want you guys to remember. During exercise, the muscle contracts and the calcium rises, which means that there's a further need of ATP and the TCA cycle speeds up. Also, in some cases, we have an IDH mutation. Okay. Another thing, it's inhibited by high levels of energy, which is your ATP and ADH. Okay, so we're talking about the IDH mutation, the isocitrate dehydrogenase mutation. In some cases, where did this red thing come from? I don't like it. It's ruining my notes. Okay, so this IDH mutation, the isocitrate dehydrogenase mutation is very high yield. It's very commonly tested. It's seen in cancers like gliomas. Gliomas was this astrocytoma in the brain, right? It's seen in gliomas, acute myeloid leukemia, and in cholangiocarcinoma. It's seen in these uh, extremely um, I'd say like it's a bad disease. Like gliomas have no cure. Acute myeloid leukemia also doesn't have very good prognosis. Cholangiocarcinoma also doesn't have very good prognosis. So on all these really, really bad prognosis disease, the IDH mutation is seen. There are two types of mutations. It can be either IDH1 or it can be IDH2, but the mutation is common. And what happens is that unfortunately, when this IDH mutation, this isocitrate dehydrogenase mutation is present, what happens is that instead of making alpha ketoglutarate, instead of making this next step enzyme, what happens is they produce 2-hydroxyglutarate, right? Instead of alpha ketoglutarate, what are you producing? You're producing this 2-hydroxyglutarate. And this is an oncometabolite, right? What is an oncometabolite? An oncometabolite is basically something that alters the DNA methylation and blocks cell differentiation, and this promotes tumor formation. This is very, very high yield. So an oncometabolite alters the DNA methylation and blocks the cell differentiation, promotes tumor formation. So it's seen in these really bad tumors, guys. It's an examiner favorite question. So an IDH mutation will lead to 2-hydroxyglutarate formation, which is an oncometabolite. It basically changes the DNA methylation. Abnormal methylation predisposes the person to develop cancer. So young patient with brain tumor is mostly an IDH mutation. It has a better prognosis than the wild type gliomas for which you don't even find a reason. And you can, in selected cancers, you can use IDH inhibitors. Remember, IDH mutation leads to production of the 2-hydroxyglutarate and oncometabolite that drives cancer through epigenetic changes. It's very high yield to know this. Okay, so we've made the alpha glutarate. Now, since we've removed the carbon dioxide molecule here and we produce NADH and hydrogen ion, we say we've done the first step of oxidative carboxylation. We now move on to the step 4 where we are taking the alpha ketoglutarate, and we're converting it into succinyl CoA. And this is done by the alpha ketoglutarate dehydrogenase, right? Dehydrogenase. We were dehydrogenizing here also, removing the carbon dioxide. We are dehydrogenizing here also, we're removing another carbon dioxide. So remember that the dehydrogenases are basically removing the carbon dioxide. Another alpha ketoglutarate dehydrogenase complex, we're removing another carbon dioxide, right? Okay. So this also produces the NADH and it produces a carbon dioxide, right? So this is also a oxidative decarboxylation step that's happening. This reaction resembles the pyruvate dehydrogenase complex that we were using to convert the pyruvate into the acetyl-CoA, and it uses the exact same 5 cofactors. Remember, we did the tender loving care for Nancy, the thymine pyrophosphate, the lipoic acid, the coenzyme A, the FAD, and the NAD plus. Do you remember that? I hope you guys remember it. If you don't, go back and look at my glycolysis part 2 video. Okay, so obviously, if you're using the alpha ketoglutarate 5 carbon molecule, converting into succinyl CoA using a succinyl CoA using this alpha ketoglutarate dehydrogenase complex, you need all those 5 factors that we were using in the pyruvate dehydrogenase complex also. So all anything that affects the vitamins that were part of the cofactors of that enzyme complex. For example, the thymine pyrophosphate was made by vitamin B1, right? That was a cofactor you needed for this dehydrogenase complex. So if there's a B1 deficiency, which means it is a thiamine deficiency, which is basically seen in alcoholics, in malnutritious people, in hyperemesis, in bariatric surgery patients. The result is less alpha ketoglutarate dehydrogenase complex activity, which means there's less ATP formed, there's lactic acidosis, and there is neurological symptoms. Also in arsenic poisoning, we studied that arsenic binds the lipoic acid. The lipoic acid is also a cofactor for this dehydrogenase complex. Therefore, it inhibits the pyruvate dehydrogenase. It also inhibits the alpha ketoglutarate dehydrogenase. The classic symptom was the garlic breath, and the medicines we were giving were the dimercaprol and caprol and the succimer, right? I've done this before, so I'm not spending too much time on this. So let's go and look at our next step, we're basically taking the alpha ketoglutarate and converting it into succinyl CoA, right? We had this tender loving care for Nancy, this thiamine pyrophosphate, lipoic acid, coenzyme A, FAD, NAD. All of these cofactors were used for the alpha ketoglutarate dehydrogenase complex. Products were the NADH and carbon dioxide. I told you we're basically removing a carbon dioxide molecule, we're creating an NADH. It uses the exact same cofactors as pyruvate dehydrogenase. So any deficiency for thiamine in alcoholics or malnutritious people, or the lipoic acid was being changed by the arsenic. Arsenic was binding it. So any of these same problems that we saw in pyruvate dehydrogenase will affect this step also. It's activated by calcium. Obviously, I told you the skeletal muscle when it's moving, it releases calcium, and that basically tells the Krebs cycle to work more, the alpha ketoglutarate dehydrogenase to work more. And it's inhibited by high levels of the NADH and succinyl CoA. So can you see how the succinyl CoA is inhibiting this? The products of the steps are inhibiting the enzymes itself. It's like this automatic feedback system, right? Okay, let's move on to the next step. We've created succinyl CoA at this step. So succinyl-CoA is done. We now need to make succinate. In order for the four-carbon succinyl-CoA to become the succinate, we need to remove the CoA. We need to remove this CoA in SH group, right? And we need to produce GTP. The GTP is the same thing as ATP. Do you remember we said that one GTP is equal to one ATP? So we're producing GTP here and we're removing the CoA SH group. Let's see how that happens. This is our step five, guys. So let's go ahead right into our step five. So step five is basically the succinyl CoA being converted into succinate. Um, and the video is getting so long. I need to literally finish this and stop the video here. The succinyl CoA is converted into succinate by the enzyme succinyl CoA synthetase. During this, we are removing the GTP. We are also removing the CoA group. The clinical correlates go something like the heme synthesis uses succinyl CoA. So when we're creating hemoglobin, when we're creating the first step in heme synthesis, the succinyl CoA is converted into this 5-ALA compound, and it's the first step in heme, right? And vitamin B6 deficiency, whenever there's vitamin B6 deficiency, if there's a deficiency in pyroxamine phosphate, it's needed for this ALA synthase enzyme. If ALA synthase is impaired because there's a vitamin B6 deficiency, there's less heme production, and you get microcytic sideroblastic anemia. And lead poisoning. Lead inhibits heme synthesis at two steps, at the dehydratase step and the pyrrokelidase. So there's less heme production. So this is very, very closely linked with heme production. And any deficiency in vitamin B6 will lead to a deficiency in the ALA synthase enzyme, which will also lead to a deficiency in your um, succinyl CoA synthase. Okay. So succinate is the only step in TCA cycle that produces GTP or ATP directly. And let's stop here because the video is getting too long. I will do the next four steps in the next video. I know the videos are long for biochemistry, but biochemistry is like it takes time, but it's not very complicated. You just need to sit and understand the reaction, and then it's not hard. Like I genuinely think it's not hard. What I would genuinely suggest is that you make these printouts of these different reactions. Like for example, you have glycolysis and you have the fate of pyruvate, and then you have the Krebs cycle happening, and you put them near your study table, and you keep on looking at them like every day. So all of the processes start making sense to you. Like you just need to do this like for a week or two. And some people also have like these really good charts you get at these bookstores for biochemistry. But that's about it. Okay, so that's it. Thank you so much, guys. I will finish the video here and start off with the next one in the next one. Okay.